Let's look at the activation and clonal selection of naive T cells. Recall that a naive lymphocyte is one that has left its primary lymphoid organ, and this is where it undergoes its maturation process, and moves into secondary lymphoid organs, but has not come in contact with its antigen. So it hasn't been activated yet. Let's look at the process by which they become activated. They have to receive two signals in order to be activated. So let's look at what happens here with the APCs and T cell activation. The first signal is that the TCR, the T cell receptor, and its CD protein must bind with the MHC antigen complex on the presenting cell. And that's what I described in the previous slide. So if you go back here, this is the first step. All of this has to bind. The first signal. The second signal is that you have co-stimulatory proteins that are expressed by the APC as well as a receptor expressed on the T cell, and these have to bind. So the co-stimulatory protein is a ligand. Remember, ligands are chemicals that bind to protein receptors, and you're going to have this co-stimulatory receptor on the T cell. You also have co-inhibitory proteins that can be expressed by APCs. We're not going to talk about those, but as the name implies, inhibitory, it would inhibit activation. If these two signals do not occur, the T cell will become unresponsive to its antigen, and it may even undergo apoptosis, cell suicide. But anergy is the term meaning unresponsive. If that antigen is a self-antigen, you do not want the T cell activated against a self-antigen. So this is just a protective mechanism. Both signals have to occur in order for activation of a naive T cell. If both signals do not occur, anergy usually happens, or apoptosis. If both signals do occur, the T cell then will be activated and it will begin to proliferate. So you'll have proliferation where it starts to divide, and it will give rise to memory cells and effector cells. That is the differentiation. Here is a dendritic cell. It has phagocytized a bacterium. It brings it in, and remember that it's going to break down the bacterium, hydrolyze it or digest it, and some of those molecules will function as antigens. The antigens then, in this case, since we're talking about an APC and a dendritic cell, expressing the antigen or presenting it to a CD4, this is going to be an MHC class 2. So the antigen is expressed on the MHC class 2. As a process of the phagocytosis, a co-stimulatory protein is going to be expressed by this dendritic cell. Dendritic cell then comes across a CD4 cell, T cell, that has the T cell receptor that recognizes that specific antigen. When it comes across that, the TCR will bind with the MHC antigen complex as well as the CD4. If this is going to become a helper T cell, this CD protein has to be a CD4. So all of these bind. That's the first signal. The second signal is the binding of the co-stimulatory protein on the dendritic cell. So you see that right here. And the CD4 has also expressed a receptor for that co-stimulatory protein, and those two bind together. So both signals have occurred. That allows this T cell to be fully activated, to begin to proliferate and differentiate, producing helper T cells, which are your effector cells, and they're going to go out and help activate other immune cells to mount an immune response, and then memory CD4 T cells that will stick around for subsequent exposure to the same antigen. So this will give you immunological memory. An interesting thing about this whole process is that Much of the research that is being done currently on immunotherapy for cancer involves looking at these co-stimulatory molecules.